Well, hi, everybody, and thank you so much for attending today. Our title of our program is Midyear Update, Next Generation Antiandrogens and Prostate Cancer, and I'm so pleased to have the opportunity to moderate this really fun program and educational program. I'm Neal Shore. I'm the medical director of START Carolinas and the head of geo-oncology for START Research in South Carolina. Very importantly, I have a fantastic privilege to work with two exceptional researchers and educators. Alicia Morgans, who is the GU Medical Oncologist, Medical Director Survivorship Program at the Dana-Farber in Boston, Massachusetts, in the US, and a dear friend and colleague, Christian Gratzke, who is a professor and chair, Department of Urology, University Hospital, Freiburg, Germany. And so we have an international faculty, we have urology, oncology, and medical oncology. So we're really looking forward to presenting with you some of the most contemporaneous information that's just come out in the last few months. We're going to be discussing these updates and how do we think best about using our second generation antiandrogens in prostate cancer. We're really going to be focusing for the most part on the metastatic hormone-sensitive prostate cancer group. Sometimes we call this the metastatic castration-sensitive prostate cancer group, our population. I think we're all quite familiar with the similarity, obviously. It's just the nomenclature. And these ARPIs, or the androgen receptor pathway inhibitors, and what have we learned over time? And how can we make sure that we're optimizing patient care? And, as often is said, we have an embarrassment of riches. So we have really 30 minutes to go through three very important MHSPC trials. We have the Aronote, which Alicia Morgans will present. Christian Gratzke will present the work, the 5-year follow-up on ARCHES, and I'll review the work from the Amplitude. All of this was presented at ASCO just a few weeks ago. There was some additional work on Aronote at the AUA. And I think it's a really wonderful time right now for all of our colleagues, whether you're in medical oncology, radiation oncology, uro-oncology, wherever you are in the world, to take a step back and say, what am I doing and how am I optimizing patient care? So with that, I'm going to hand it over to my good friend and colleague, Alicia Morgans, to talk about Aronote. Thank you, Alicia.
Thank you so much, Neal. And I was really pleased to actually be part of the presentation of the Aronote data update of quality of life outcomes at ASCO just a few weeks ago. So Aronote, just to remind everyone, was a phase 3 study that was designed to include patients with metastatic hormone-sensitive prostate cancer, and patients were randomized 2-to-1 to receive ADT with darolutamide or ADT and placebo. And the purpose here was to determine whether the combination of ADT and darolutamide would be superior when compared to ADT and placebo, with the primary endpoint of radiographic progression-free survival, because we already knew that combining ADT and docetaxel with darolutamide was going to be superior to a chemohormonal backbone alone. So as we can see here, RPFS was the primary endpoint, and there were multiple secondary endpoints, which included quality of life, which I'll get to in just a minute. When it came to that initial readout, there was a 46% reduction in the risk of radiographic progression or death. And in terms of the secondary endpoints, there was significant benefit across all of these, including that time to metastatic castration-resistant prostate cancer, pain progression, PSA progression. And really, the overall survival data was immature, and it was not powered to assess this particular outcome. We did see that PSA responses were more pronounced among patients who received ADT and darolutamide. So 62% achieved that PSA of less than 0.2 versus 18.5% with placebo. And the safety profile was similar between the arms.
So, Alicia, can I ask, I know you have another slide to present. Recently, Christian and I were at a program together in Germany, and one of the questions that was asked was, really appreciate this hazard ratio of 0.54 on the RPFS, the primary endpoint of the study. OS was early. And we may not see an OS in this trial because of all the other agents that are available for crossover. And we're going to talk about some 5-year OS data in a second with the ARCHES study. How do you respond to someone who says, yeah, I see this really provocative RPFS benefit? Now, I guess, you know, we know the OS data, significant OS benefit from Aramis and Arasens, but how do you respond to that inquiry?
Yeah, I would say this study was actually a pretty small one when it comes to understanding overall survival. And the other trials were really designed as much larger studies with planned longer follow-up. And this was really designed around short, sweet, let's get the RPFS endpoint. And I don't think we'll see any further outcomes because the study was stopped when it essentially reached that primary endpoint. Also, and I confirmed this, when the study ended, when they reported that primary data, patients who were on the control arm could receive treatment with darolutamide. This was built in. But of course, patients in the regions where this was performed could also receive AR pathway inhibitors on progression. And to your point, this study being a more contemporary study than the studies that we now have as really backbones cementing this ARPI plus ADT combination as being standard of care, didn't have access to all of the many things that patients on this particular trial are getting and have gotten. So I think there's multiple reasons why this is going to be difficult, but especially, I mean, if you look at that control arm, 223 patients, that's very, very much smaller than any of the other control arms on these trials. And I think, especially if we're looking for something like overall survival, we need long follow-up, we need large studies, and this was really not designed to be that way.
And of course, we saw OS benefit in Aramis, the NMCRPC, and Arasens in the triplet. So we know there's OS benefit. Christian, can I ask you, what do you make of this PSA response? You know, really 2/3 of the patients with the Daro arm, the combo, getting a PSA nadir of less than 0.2 versus less than 20% in the ADT placebo getting under 0.2. And we even at AUA, we presented some data on ultra low PSAs, less than 0.02, 5 times more likely in the Aronote combination group. What do you think about that? How are you utilizing PSA nadir?
I think this is a really impressive and very impressive response rate, showing that the drug is actually very efficacious. And to your point that you made before, I think this is a very good answer to somebody who questions the efficacy of the drug, if any does that. And it also cements the fact that ADT monotherapy in those patients and that clinical scenario is really not sufficient. But the efficacy and the good drop in PSA here is a very good sign of the efficacy of the drug.
Yeah, I love the point you just made is, you know, monotherapy ADT, low volume, high volume, synchronous, metachronous, de novo recurrent. There's a very, very limited role for that. But so, yeah, let's see what happened there. We kind of.
Sure, I'm happy to go through the quality of life data. So thank you for that, Neal, and great conversation. I think this data also suggests that the drug really works. So when it comes to the comparison between the two arms in terms of quality of life, both overall and in terms of pain specifically, ADT and darolutamide was favored over ADT and placebo. So as we can see here, time to pain progression was delayed when we used ADT and darolutamide versus ADT and placebo. The hazard ratio here is 0.72. The time to the deterioration or decline in the quality of life of patients was extended when we added darolutamide. So it actually prolonged that time to deterioration by about 5 months versus ADT and placebo, which I think is long and meaningful and really interesting too. I think when it comes to the next issue, this comes to the points that were being made around those low PSAs. We saw that patients who reached a PSA of less than 0.02 or less than 0.2 during their follow-up actually had a longer time to pain progression and a longer time to deterioration of their overall quality of life versus patients who didn't have such a pronounced response in terms of that PSA, really suggesting that we can actually improve cancer control and improve quality of life very meaningfully with this combination. And then the overall well-being showed that the specific domains that seemed most improved with darolutamide included social and family well-being, as well as functional well-being, the ability to get around, go out, do the things that make you happy. And also, really interestingly, urinary symptoms specifically were improved.
Yeah, I really, have great admiration for the work that you've done on these patient-reported outcome work, this health-related quality of life outcomes. This is very important data, Alicia, and you've really been, at the top and championing this to help us better understand how we make decisions. And we talk about this embarrassment of riches. We've got abiraterone and enzalutamide and apalutamide and darolutamide. Christian, how do you, when you're in the clinic and you've got a very busy clinic and you're working in a multidisciplinary team with your medical oncologist and yourself, how do you address this with patients, all these different types of health-related quality of life concerns in addition to the clinical benefits?
Well, I really, oh, did you say Christian? I'm so sorry.
I was going to
go ahead.
But you go ahead, Alicia, and then I want to hear Christian too.
I apologize. I misheard, and I would actually rather hear Christian. But I'll just simply say, just briefly, that I usually kind of dig into each of these quality of life aspects and try to find the ones that are specifically bothering that person, and then try to work with them on whether it's going to be the ADT ARPI combination. It's usually not. It's usually all the supportive things that we do around it, but trying to make sure that those are optimized as well. And go ahead, Christian. Apologies.
Well, I think for me, when I talk to my patients and I explain them the options that they have before I start a certain treatment regimen, they would like to know, first of all, about the efficacy, but certainly about the quality of life and whether or not the drug has a specific negative impact on quality of life. And if I can tell them that over a certain period of time, they won't experience a negative impact on quality of life. And in that case, Alicia, you showed, and I really liked the way you portrayed that, even less urinary symptoms. Of course, that would need to be detailed, but certainly the patients can expect a time where he can lead his normal life, has a good clinical benefit, but leading his normal life the way he wanted it before the start of the disease. And that's, I think, very, very impressive for patients to hear. And it certainly makes him go on that drug with a strong belief that it actually works.
Yeah, well, thanks so much. And in the U.S. now, based upon the Aronote study and the JCO publication, it's now approved by FDA and in the NCCN guidelines. So great work, Alicia, and thanks for that presentation. Let's go over to ARCHES now, Christian, if you could review that for us, please.
Thanks, Neal. So this is the ARCHES trial, and it shows that specific abstract shows the 5-year follow-up of the overall survival analysis. The study design is similar to the one that Alicia just showed us. 1,150 patients, also with MHSPC, who were randomized into receiving placebo and ADT, or enzalutamide and ADT, which is another novel hormonal agent, or RP as you call it. The primary endpoint in this trial was the radiological progression-free survival, and you can see a variety of secondary endpoints, such as overall survival, PSA, progression-free survival, and also one very important parameter that you just heard about was safety. So this 5-year readout had a median follow-up range of 61 months. And with regard to overall survival, it showed that patients who were treated with enzalutamide and ADT versus placebo and ADT had a significantly prolonged overall survival with a hazard ratio of 0.7. If you look at this by disease volume, Enza increased the overall survival in high volume patients versus placebo by about 3 years, which is very similar to what you saw in the low volume patients. In other words, after 5 years of treatment, the combination of enzalutamide and ADT was leading to a survival benefit of about 3 years, which actually is impressive. With regard to safety profile, we saw that the long-term survival improvements were accompanied by a slight increase in emergence treatment emergent negative effects, but these were diminishing over time. And they were very much in line with what we knew from the same drug in other previous indications.
Yeah, that's really, kudos to all the folks and the sites globally who participated in ARCHES. I think likewise, there was recently a long-term follow-up with Enzamet, mostly an Asia Pacific study, a little bit different, had some different inclusion criteria. Comparator arm was an NSAID, and there was some prior more significant use of docetaxel, but again, demonstrating the OS benefit. And so, but again, these studies were done many years earlier and begun many years earlier, as opposed to Aronote. But again, it's a great accomplishment to see this sort of 5-year data. Alicia, based upon what Christian presented here in this nice presentation that Andy Armstrong did, the publication is pending. I'm sure it'll be in a high impact journal. How do you think about the fact that now enzalutamide has been around and has this durable demonstration of survival benefit, and the safety profile is pretty well established?
Well, I think it gives us a great option for treatment in this metastatic hormone sensitive setting. I think, when we're trying to work with patients to find the best treatment for them, first, of course, I often consider, do I do a doublet or do I do a triplet? Enzalutamide is not typically one that I include in my triplet combinations, just because fewer patients in the Enzamet trial kind of had that clean randomization, the way that things happened in PEACE-1, a little bit cleaner, and Arasen's much cleaner. But if I'm using ADT and an ARPI, enzalutamide sometimes comes into those conversations. And there are certain situations where it's so helpful for a patient with liver function enzyme abnormalities. As an example, there's no dose reduction, and this can be really important. So, I think there's always room to have more options, and it's always reassuring for us as clinicians and for patients to see this. In general, though, too, I think the message here that ADT and ARPI can give sustained benefit. And I think about 33% of patients still on therapy without progressing at this 5-year mark. That's hugely important to patients. And I think regardless of the specific partner that I use with ADT, I like to share that message because I think it's really hopeful and encouraging to everyone in the room that this can be a really, really helpful treatment.
Yeah, thank you for that. Christian, Alicia brought up an interesting, mention of triplet versus doublet. So in Germany, maybe you can even speak for Europe now. What are your thoughts on, the triplet versus the doublet decision for patients who present with a newly diagnosed MHSPC, maybe perhaps they're chemo fit, high volume. It's always a controversy. Do we add those 6 cycles of docetaxel or not? How do you think about that?
So the way I talk about this with patients is the fact that if you have a very aggressive disease, you would like to attack the cancer cell by various mechanisms of action. So if you have a patient that you feel he is a good candidate for docetaxel, that means that you can maybe improve that situation by adding to the doublet chemotherapy. The question whether or not the patient is applicable for chemotherapy is an individual decision, which needs to be discussed with the patient in detail. And it's dependent on various factors. It's ultimately a decision the physician takes in the communication with the patient. And I'm a very big supporter of triple treatment. If I feel that the patient has a treatment need and wishes to have the most thorough and most efficacious treatment that is available. Now, there's always a discussion whether doublet or triplet is better. And it depends also on the region where you live in. But based on the facts and based on the Arasen's trial that was presented earlier, the triple treatment is a very efficacious treatment options in patients that wish to have that.
Yeah, thank you. Alicia, considering this, proverbial embarrassment of riches now, 9, 10, 11 to possibly even 12 trials now. And we're going to present another one in a second regarding the options other than monotherapy ADT. What have you learned, and what would you suggest? And maybe, Kristen, you can also weigh in on this. How do we improve the multidisciplinary team efforts for making these optimal decisions for patients?
Yeah, I think it's interesting because I don't even talk to patients really about ADT alone. We are always talking about ADT and something. And that's not to say that there isn't a situation where someone doesn't do well with single agent ADT and I have to then kind of manage that situation and don't really have the opportunity for many months or perhaps ever to add on an ARPI. That's rare, but sometimes that happens. I think in general, we approach it as you're going to start your ADT. Within a month, have you back and we'll go over safety and tolerability. We'll optimize the situation. We'll plan to add something else to it and we'll get better disease control. And it's never really an ADT alone kind of a conversation. And my nursing team reiterates that and talks to the patient in between visits to make sure that they're not having trouble with hot flashes, insomnia, mood changes, and other things that we might be able to intervene upon and otherwise. And the other thing that we really focus on from a multi-D perspective, everybody kind of saying the same message, is physical activity, because physical function is improved, mental health is improved, and it's extremely effective. The most effective thing of anything I could do, I wish I could make it into a pill. Physical activity and exercise really seems to help.
Yeah, I agree. Kristen, any additional thoughts?
Just echo what Alicia just said. There's two distinct messages from these two abstracts. You have a preservation of quality of life, and you have a very good efficacy over a long period of time. And if this comes up, there's no conversation about ADT alone. I very much agree with Alicia here.
Okay, thank you so much. All right, let's go to our next, our third presentation, and this was wonderfully delivered by Gerda Tard at ASCO, and this is the first of other triplet therapies now called the Amplitude Trial. So this is a large global trial that looked at abiraterone, acetate prednisone, and niraparib versus ADT and abiraterone, acetate, and prednisone. And of course, ADT in the triplet arm for abiraterone and niraparib. And these, again, were in patients with low volume and high volume, de novo, recurrent, MHSPC. And they had at least one of or more alterations in homologous recombinant repair genes. And you see them listed here. I believe it's about 9. And they had, for the most part, the overwhelming majority had ECOG performance status of 0. There's a 1-to-1 randomization, and you can see it in the study schema here. And so we've learned a lot from a prior trial with niraparib called the Magnitude trial. We have had 2 other phase 3 trials in the resistant population, the PROPEL and the TALAPRO-2 trials, looking at different versions of PARP inhibition. And what we've seen in the approval of those 3 resistant trials was saying, okay, well, what happens if we now look to the castration sensitive or the hormone sensitive patient population? That's exactly what was done here. And so you see the exclusion criteria and some other prior permitted treatments. It's a 1-to-1 randomization, a fairly good sized number of patients here, about 700 total. And the primary endpoint was rPFS, investigator reviewed. Secondary endpoints. The bottom line was that, this was a successful trial with an approximately hazard ratio for the intent to treat of these 9 genes of about 0.6, even lower to about 0.5 on the BRCA2, BRCA1. There was really no unusual adverse events on the safety profile. The nice thing here, what we see is less than 5% a discontinuation rate. And that's actually good. Of course, these are younger patients than in the resistant population. And also, because of that, most likely a better performance status. I think the adverse events that were documented and reviewed by Gerda Tard were consistent with the class effect of PARP inhibition, a need to monitor for myelosuppression and some GI toxicity. So the conclusion was that niraparib and abiraterone acetate prednisone in conjunction with ADT improved the outcomes for these patients with HRR gene alterations. There was a subgroup analysis. It really was the predominant benefit was in the BRCA2 and somewhat the BRCA1s. And that was a key subgroup analysis. I think what this really opens up is a lot of opportunity to think about, how are we doing our genetic testing? Are we checking, how it varies around the world? Are we doing germline testing, somatic testing? But this is, again, this conclusion of a biomarker precision-based, sort of better tailored based therapy based upon genetic testing. I think it's too early to see an OS benefit, which is not surprising. But this is, I think, going to be one of the first of several other MCSPC PARP combination trials that will continue to read out. So let me ask you, Alicia, how did you feel when you saw this data? Were you surprised? And how do you think this will come to a potential regulatory approval?
I was really excited. I think, this is the first in this early phase where we really saw rPFS, this signal for OS maybe going sort of in the right direction, just immature data. But I feel like we have wanted to tackle these BRCA mutations in as early setting as possible. And this was the first opportunity, the first data that really helped us to understand if that was possible. Safety signal looks reasonable. We need more follow-up information here, but I feel like this is going to be moving these agents into this early phase, as long as it's safe from a long-term perspective, as long as we understand how to care for people, will be so important for people with some of these high-risk mutations. Though we may find that we prioritize things like BRCA2 with these kinds of treatments rather than others. I think that still needs to be determined, but it's, I think, very exciting.
Yeah. Christian, your thoughts on this Amplitude data?
Oh, very much like Alicia's thoughts. I think that this personalized biomarker-driven approach is something that we definitely need, especially in this early setting. What I'm specifically interested to know is the fact that we know that BRCA mutated patients do very well. But if you look at the other, the non-BRCA alterations, there were some specific alterations like PALB2, without going into too much detail, that we thought might allude to a very significant clinical benefit. But it turned out not to be. But then again, very few patients did have these very, very rare alterations. So I think that's for the future to figure out who are these patients that benefit best from the use of those PARP inhibitors at this very early disease setting.
Yeah, extremely well said. Thank you both for that. So we always kind of love this slide, right? Because it just speaks to the amazing advances that have happened in the last 20 years. Certainly all homage to Huggins and Hodges in 1944 showing castration, but that's no longer the standard of care, whether it's in the sensitive population, the non-metastatic, the BCR population, the MHSPC population, and clearly in MCRPC. This one study here that recently came across, which I think is very importantly done, is this combination of radium and enzalutamide, the Peace 3 trial, and kudos to the EORTC for getting this across the finish line. Christian, comments about this and in using two different novel agents, as you just said earlier. That's one of your tenets for how you think about things.
So this is highly interesting because the use of radium comes into play now at a specifically later time point. We didn't see too many trials on radium over the last couple of years. So now to see this combination with Enza leading to a significant survival benefit is interesting. We can discuss which patients will benefit from this combination the most, but it certainly helps us to augment our armamentarium to treat our patients in that disease setting.
Yeah, absolutely. Alicia, any other comments regarding that? I know it's so open-ended, but it's really an exciting time now in both sensitive and resistant populations.
I would agree to echo the congratulations to the team for making that happen, and also to point out that this was really an asymptomatic, minimally symptomatic patient population. They had lymph node involvement. They didn't have visceral involvement. So it almost expands, or does expand, the group of patients in whom we might consider something like the addition of radium.
Well, thank you so much. I think we're really right up at time. This is so much fun and enjoyable for me. I could have gone on for a few more hours on this. It's always great to end on a slide that keeps the audience invigorated. There's so many more trials that are out there now, phase 3s and some very provocative phase 2s. Alicia Morgans and Christian Gratzke, thank you so much for your expert work and your insights on this. It's really been great. I hope the audience found this valuable. I really appreciate everybody's time and look forward to seeing you at upcoming congresses. I think the next one we'll look forward to seeing everyone at is ESMO 2025 in Berlin. So thank you very much.
Thank you.
Thank you very much.